Hello students, I'm Atul Kumar Singh and today again I brought for you class 12 subject biology chapter 2 sexual reproduction in flowering plants. It is the part 2 of this lecture, this chapter. It is totally based on NCERT. So let us start. Here we are going to discuss the pollination. First thing we are going to discuss the pollination. Before this, I'm going to tell you that I have completed the part 1 of this chapter. So please, if till now you have not watched that video, please visit it. So you will be able to understand the next part of this video. So now see, transfer of pollen grain from anther to stigma. This is known as pollination. Or if you want to define it, it is very simple definition. How the pollen grains will be moved from anther to the stigma. This is the very simple definition. Okay. Here you can say transfer of pollen grain shed from the anther to the stigma of a pistil is termed as pollination. Now, how many types of pollination can be? So if you want to know this thing, you have to keep three things in mind. One thing, if the anther and stigma are present on the same flower, number one condition can be there, but the anther and the stigma both are present on the same flower. So if this is going to happen, it is known as autogamy. See, self-pollination. Here what is going on? Transfer of pollen grains from the anther to the stigma of the same flower. That means plant is also the same and flower is the same. Okay, so here what is going on? It is just like a self-pollination. What will be the thing over here you have to keep in mind? That is synchronization. Synchrony requires. What do you mean by synchrony? Actually, synchronization means at the same time stigma and anther, that means I should say pistil and what you call stamen, both should be mature at the same time. So in the stamen, anther will also be ready, pollen grains will be ready, and at the same time the stigma style ovary will also be ready. So what will be there? Anther will release the pollens, these will come to the stigma, and the stigma will accept it. So this is just like self-pollination. It is what? Self-pollination. Now what are the example? You can say that viola is the example, oxalis and commelina. Here you see the production of two types of flowers will be there. Usually two types of flowers. One is chesmogamous flowers. Chesmogamous flowers are just like similar to normal flowers, which expose the anthers and stigma. That means they, here actually anther and stigma both are exposed. So here what can be there? Here actually cross pollination can also be there. There is a chance of cross pollination. Okay, chesmogamous flowers and cleistogamous. Second type is cleistogamous. So here these do not open, these flowers do not open at all. So there is no cross, there is no chances of cross pollination. It will be completely self pollination. It will be completely self pollination. So dear students, you must know autogamy is a type of self pollination where transfer of pollen grain from the anther to the stigma of the same flower will be there. And in case of few plants, two types of flowers can be there. One is chesmogamous flowers and other is cleistogamous. In case of chesmogamous flowers, self pollination can also be there and cross pollination can also be there, because these are open, just like normal flowers. In case of cleistogamous flowers, in case of these, since these do not open at all, so these cleistogamous flowers always undergo autogamy or self-pollination. So is it clear? If you will have any doubt, please mention in comment section. I will clear your doubts. Now, second point I want to tell you is the zetonogamy. In case of autogamy, you saw that pollen grain from the anther of the same flower is transferred to the stigma of the same flower. Understood? Stigma of the same. So here flower is the same, but in case of zytogamy, flower is different. Okay, so it is a type of cross pollination which is functionally cross pollination, but actually it is not, because the plant is the same. What is the same? Plant is the same. So now see, another flower, here flower is changed, another flower of the same plant, another flower of the same plant. Now see, here pollinators will be required, but it is just similar to the autogamy because plant is the same. Now once again I am going to tell you this thing. In case of autogamy, plant is the same and flower is also the same, but in case of zytonogamy, plant is the same but flower is different. Now we are coming to the xenogamy. This is actually cross pollination. And why this is actual cross pollination? Because here flower is also changed and plant is also changed. So you will say transfer of pollen grain from one plant to another plant. That means pollination, what you call, here the pollen grain will move from the one flower of a plant to another flower of another plant. So I think this is clear, and this leads genetic variations. This leads genetic variations. So now, agents of the pollination or pollinators. What are the pollinators? I want to tell you, students, actually pollination will be proceed by different agencies. So what are the agencies which can proceed the pollinations? So here you see pollination by wind, pollination by wind. This is known as anemophily. What is it? Anemophily. It is called anemophily. Okay, now it is the most common method. I should write here most common, not more common. It is most common. And you must know here, pollens are light, non-sticky, on well exposed stamens, and these are present on well exposed stamens. So why the pollens are light? Because if they will be heavy, they cannot move with wind. So if they are lighter, they can easily move with the wind current. Okay, now these are non-sticky. If they will be sticky, they will stick somewhere, and after that they will not move. So they should be non-sticky, so they are non-sticky. Now they are present on the well exposed stamens. Well exposed stamens, what do you mean by this? Actually, if the stamens are not exposed, if the stamens are not properly exposed, then they cannot come in contact with the wind current, so it will be difficult for them. So here what will be there? They are well exposed stamens, and the examples are grasses, and one very good example is pinus. If you are appearing in NEET exam, this example is asked again and again, pinus. So you must know pinus is anemophilous plant. Anemophilous plant means where the pollination by wind will occur. Now, second thing we have to know, that is pollination by water. Pollination by water. So here this thing is known as hydrophily. This thing is known as hydrophily. What will be there? Mostly ribbon-like pollens are present. Ribbon-like pollens. Okay, and these are covered by mucilaginous covering. Why this mucilaginous covering is required? This is required for protection. This is required for protection. Now, what are the examples of it? So the examples are Vallisneria, Hydrilla, Zoystera. Vallisneria, Hydrilla and Zoystera. Some other also. Here I want to tell you, Vallisneria and Hydrilla are freshwater plants and Zoystera is a marine water plant. Now, pollination by animals. This is the third category and most interesting also. This is known as zoophily. This is known as zoophily. Here you must know flowers are large, okay, colorful and fragrant, okay, and rich in nectar. Dear students, these will be large, so easily the insects and all can see it, number one. Second thing, they are colorful, so colorful means the insects can easily be attracted, and they have fragrant. By the fragrance means smell. So by that fragrance or smell, good smell actually, so by that good smell, the insects will be attracted, and they are rich in nectar. Okay, so by this nectar the insects and other what you call animals can get the food, so they will be attracted. So in this manner, acha, one important thing is they are sticky. They are sticky. Sticky means here the pollen grains will be stick on the animals, so animal body, so easily they can be transferred from one what you call anther to stigma. Now, how many types of zoophily can be? So here first thing, zoophily can be by insect. Number one, insects can be one agency. If the insect is agency, we used to say entomophily, like honeybee. Next, it can be by ants. Ants, if ant is involved, we will say myrmecophily. If bird is involved, we will say ornithophily. If snakes are involved, we will say ophiophily. If bats are involved, we will say chiropterophily. And if mollusks are involved, we will say melicophily. So you may say entomophily, myrmecophily, ornithophily, ophiophily, and chiropterophily. These are the various types of pollination under zoophily. Dear students, these all are asked in NEET exams. So please learn it. Now, interesting facts I want to share, that is, in some species floral rewards are in providing safe places to lay eggs. Okay, an example is that of the tallest flower of Amorphophallus. The flower itself is about 6 feet in height. Okay, so here what will be there? Floral rewards are providing safe places to lay eggs. That means here few animals or insects lay eggs inside the floral parts. Okay, so that is the good example is Amorphophallus. Now, one more example we see here, that is yucca plant and moth. The moth is what you call it, smaller than insect, so that deposits its eggs in the locule of the ovary, and the flower in turn gets pollinated by the moth. That means yucca gets pollinated by the moth. Yucca plant will be pollinated and the moth will get the place to lay eggs. Now, the larvae of the moth come out of the eggs as the seed starts developing. Okay, as the seed starts developing, at that time these eggs, these larvae will come out from the eggs. Okay, next thing, outbreeding devices. First thing I want to tell you, that is what is inbreeding and what is outbreeding. Actually, self-pollination is a type of inbreeding where plant and flowers are the same. Okay, and what is outbreeding? If cross pollination is there, that is outbreeding. Cross pollination, outbreeding, and self pollination, inbreeding. So continued self pollination result in inbreeding depression. I want to tell you, if continuously for so many progenies the self pollination, repeats, then what will be there? Self pollination, if again and again it will be repeated, then inbreeding depression occurs. What is this inbreeding depression? Dear students, inbreeding depression is loss of vigorosity or loss of yield in the plant productivity. So this is the inbreeding depression. Now, flowering plants develop many devices to discourage self-pollination and to encourage cross-pollination. Okay, if the productivity will be reduced, so what plant wants? Plant want that self-pollination should be discouraged and cross-pollination should be encouraged. Okay, for this there are some outbreeding devices. So we have to now learn these outbreeding devices. See, number one device is the unsynchronization. Unsynchronization means what? If the anther and ovary is not maturing at the same time, this will lead the unsynchronization. Okay, here, and suppose the anther is matured fast and ovary is not ready. Okay, this condition will be known as protandry. This condition will be known as protandry. Okay, here pro means primitive, andry means androecium. That means androecium matures before what you call carpel and all. That means before ovary. Ovary is not matured. Stylostigma of ovary is not matured, and androecium is matured. So this condition is known as protandry. Okay. So obviously anther is ready for releasing pollen, but since stylostigma ovary is not ready, so fertilization or self-pollination cannot occur. Now, second thing is protogyny. This thing is what pro means primitive, means before, and gyne means gynoecium. Here, gynoecium matures before the androecium or before the stamen and anther. So this condition will also lead what the unsynchronization, or here also it will prevent the self pollination. Now, second agency or second device can be self incompatibility. Okay, dear students, it is a genetic mechanism by inhibiting the pollen germination or pollen tube growth in the pistil. So here two things are important. One is genetic mechanism, it is a genetic mechanism, and second thing, it inhibits pollen germination on stigma. Okay, pollen germination or pollen tube growth in the pistil. So these two things you must know. Number one, it is a genetic mechanism, and second thing, it inhibits pollen germination on stigma or pollen tube growth in the pistil. Okay, so now third device can be production of unisexual flowers. Okay, what will be there? This unisexual flower can be also a prevention in self-pollination. How? Dear students, if in a flower only anther will be present or only stamens, that means male reproductive organs will be present and female reproductive organs are absent, then also it cannot be possible. It can't be possible that self-pollination cannot be possible. Only cross-pollination will be there because there is no female at that time, female reproductive organ at that time. Okay, so this will be the thing. Next thing, one more important thing I want to share with you, that is monoecious condition. Monoecious condition, dioecious and monoecious condition I have told. Mono means single and ecious means home. So here you can understand, if a single plant has both type of male and female reproductive organs, this condition will be known as monoecious condition. But the flowers are different. Here in case of monoecious condition, the flowers will be different. Like I'm telling you, suppose one flower is having stamens only and another flower is having pistils only. So one flower which is having stamen, that will be the male flower, and the flower which is having pistils only, that will be the female flower. So these flowers are different, but suppose these two types of flowers are present on the same plant, then this will be monoecious condition. Here, what did they think? Male and female flowers are different, but these two types of flowers are present on the same plant. So this condition will be monoecious condition. So here it is mentioned that monoecious condition prevents autogamy. It can prevent the autogamy obviously because flower is having only single type of reproductive organs. So there is no chances of cross pollination. Acha. One more thing, but dioecious condition, do you know? Dioecious condition means male and female flowers are different and they are situated on the different plants. So this dioecious condition prevents autogamy and zetonogamy both. Okay, I believe that you know about the autogamy, where the flower is the same and this flower means plant is the same and flower is the same, and in case of zetonogamy, plant is the same but flowers are different. Okay, so this already you know. Now, pollen pistil interaction, this is very important topic in this chapter. So see, Pollen pistil interaction. All the events from pollen deposition on the stigma until pollen tube enters the ovule are together referred to as pollen pistil interaction. Okay, that means from here to the entry into the ovule, the whole thing is known as pollen pistil interaction. Okay, one very important thing you must know is the pistils has ability to recognize the compatibility of the pollen and accept only the right type of pollen. You must know this thing. Actually, if on rice plant, suppose there is a rice plant, wheat pollen falls on the stigma of it. Wheat pollen means on rice, suppose the wheat pollen falls on the stigma of wheat, then what will be there? On wheat, this pistil will reject it. It will recognize that this is not the right, this is not of right type, so it will reject it. Okay, so what will be there? That rejection and acceptance is the ability of the pistil. So you must know pistil has the ability to recognize the compatibility of the pollen and accepts only right type and promotes post pollination events Okay, now if the pollen is of wrong type, just like I gave example of rice and wheat, so here what will be there? Incompatible, or it is incompatible, its germination and tube growth will be prevented. Okay, it cannot germinate in this manner, just like it is germinating, so in this manner it will not germinate. Okay, this interaction is a type of chemical process. Now you can see here, stigma is germinating and it is entering, sorry, pollen tube is germinating and it is entering into the ovule. Now here few things are shown. Okay, you see, just like this point here, filiform apparatus is the apparatus which helps the entry of pollen tube inside the synergids, inside the ovule I should say, and finally it goes to synergids. Okay, now this thing we will discuss later on in detail. Now pollen germination, how this pollen tube germinates, this thing we have to see. Pollen tube germinates through the germ pore. Okay, now what is the germ pore? I told already, in every pollen actually there is a point at which, or there is a pore at which sporopollen is absent. I have told already in the first video of this chapter, so in that you can go through it, and in that you can see, or in the pollen there will be a position, there will be a point where there will be no sporopollen in, so that point is known as germ pore. So from this germ pore pollen tube germinates. Is it clear? Now contents of the pollen grain move into the pollen tube. That means what is present inside the pollen grain, that move into the pollen tube, that come into the pollen tube. Now pollen tube grows through the tissues of the stigma and style and reaches the ovary. Okay, now this pollen tube grows through the tissues of stigma and style and reaches the ovary. You can see this chapter once again. See here, the pollen grain is falling on the stigma and reaching to the ovary in this manner. Okay, so this thing is going on. Now in two-celled pollen grains, if the pollen grain has, I think you know about the pollen grain, I have already told you about the pollen grain, that pollen grain will have in this manner one vegetative cell and one generative cell. I told already, just like this is the generative cell and this is the vegetative cell. Okay, so this thing I have already cleared. Okay, now see. So here in the two-celled condition, the generative cell divides and forms two male gametes during the growth of the pollen tube in the stigma. In three-celled pollen grains, The two gametes are from the beginning. Okay, now what I'm telling you here, in the generative cell, if it is already divided, then it must have two nuclei. If it is not divided, then it must have only single nucleus. So if it has single nucleus, automatically this nucleus should undergo division and this nucleus should produce two nuclei. Okay, that means two gametes. So that production of two gametes, two male gametes, will undergo in this generation, what you call, during the germination of pollen tube. Okay, this will be there. Now pollen tube, after reaching the ovary, enters the ovule either through micropyle, this thing is known as porogamy, or through chalaza, this is known as chalazogamy, or through any other place, that is known as mesogamy. Okay, usually by micropyle. So you must know, usually by micropyle. Now, and then it enters one of the synergids. That means pollen tube enters into synergids through the filiform apparatus. This thing I have already told you. Filiform apparatus guides the entry of pollen tube. Filiform apparatus guides the entry of pollen tube. Now, bagging, tagging, emasculation and artificial hybridization. This I am going to tell you. Dear students, emasculation is the process of removal of anthers from a flower. That means if a flower is bisexual means it has, suppose, male and female both types of reproductive structures, then suppose it has male and female reproductive structures, then we remove the male reproductive structures, that is anthers. We will remove the anthers. So the process of removal of anthers from a flower is known as emasculation. After that, we put a polybag on it, that is called bagging. Either polybag or paper bag can be there to prevent the what you call falling of pollen grains from a different flower. Okay, to prevent it, we don't want that another flower, another pollen grain should come and it should fall on this type of flower which we have emasculated. So we will just put the poly bag or paper bag on it. This is known as bagging. And we will do tagging. Tagging means we will write the name of the what you call breeder, that means name of the scientist who is doing this, or just like if you are doing, then you will put your name, that I did it, and the date of a, like a particular date should be there, so that date you will mention, and what purpose it is, that also you can mention. Experiment number you can mention, observation number you can mention, all these things you will mention over here. Now, why you did it? Because you want that this flower should undergo cross pollination, so for that purpose you do it. So here what will be there, if you will do cross pollination, so by cross pollination you will get a hybrid plant. So this process will lead the artificial hybridization. So here what will be there, by this cross pollination, when it will be mature, you will do the cross pollination of it, so that cross pollination leads the production of a hybrid fruit, I should say, or hybrid seed, I should say. So that thing is known as artificial hybridization. You will study this thing in what you call higher classes. Now, double fertilization. We are coming to this part. This is very important in angiosperms, and you must know. Double means two times. Fertilization means fusion of male and female gametes. So here, the fusion of male and female gametes two times is known as double fertilization. We will see here. First thing, it is discovered by Navashin. Second thing, the pollen tube releases the two male gametes into the cytoplasm of the synergids. I have already told you that pollen tube will have one vegetative cell, one generative cell. Generative cell undergo division if it is having a single male gamete. If it is having two male gamete, there is no problem. But if it is having a single male gamete, it undergoes division and it will produce two male gamete. Ultimately, generative cell must have two male gametes. So these two male gametes will enter into the synergids. these two male gametes will enter into the synergids. Now, one of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. Dear student, it is the important process for production of eggs and seeds. Okay, here what will be there? Two male gametes are present in the generative cell. Now, one male gamete will fuse with the egg cell. One male gamete will fuse with the egg cell. So, male gamete plus egg cell. This fusion will be known as syngamy. This fusion will be known as syngamy. Okay. And here, what will be produced? So, this results in the formation of a diploid or 2n zygote. What will be produced? Zygote will be produced. Zygote will be a diploid cell. Okay. Why it is diploid? Because one male gamete and one egg cell meets, so it is a diploid. Now, the other male gamete moves towards the two polar nuclei located in the central cell. Okay, and fuses with them to produce a triploid 3n primary endosperm nucleus, that is PEN, primary endosperm nucleus. As this involves the fusion of three haploid nuclei, it is termed as triple fusion. Dear students, once again I am telling you what will be there. The other male gamete means one male gamete is employed over here, and other male gamete is fusing with two polar nuclei. It is going to be fused with two polar nuclei which are located in the central cell. So here two polar nuclei plus one male gamete, this one. So overall how many will be there? 1 plus 2, that means 3. So it will be producing a triploid, that means 3n, primary endosperm nucleus. So this primary endosperm nucleus will be a triploid structure. Okay, so it is clear. Now this process, since it is triploid, so this type of fusion is known as triple fusion. It is known as triple fusion. Okay, now hence you saw that two types of fusions are there. One is syngamy and another is triple fusion. Since two types of fusion are there, that's why it is known as double fertilization. It is known as double fertilization. So it is clear. Now double fertilization means what? Double fertilization means syngamy and triple fusion. Syngamy and triple fusion. So dear students, I know you may be confused, but please watch this video twice or thrice. Everything will be clear. Okay. Try to understand these things. Now, the central cell after triple fusion becomes the primary endosperm cells, that is PEC. Okay, because central cell will have one nucleus which is a triploid. That nucleus is known as primary endosperm nucleus, but the cell is known as primary endosperm cell, and it develops into the endosperm. Okay, while the zygote develops into the embryo. Actually, the product of syngamy, that is zygote, and the product of triple fusion, that is primary endosperm nucleus. So that primary endosperm nucleus will produce endosperm, and the zygote will produce embryo. Okay, so it is clear now. This is showed in the diagram. What is going on here? Just see. This is the zygote, and this is the this red colored. This thing is what? This thing is primary endosperm nucleus. Okay, what will be in zygote? First you see the zygote only. Here this is the zygote, which is having a single diploid nucleus, just like from here I have shown. This zygote undergoes division, so two-celled condition will be found, then such type of condition will be found, and then such type of condition will be found, and then such type of condition will be found. So here you see one-celled to two-cell, then two-celled to such type of globular embryo will be found, and then such type of heart-shaped embryo will be found, and then such type of mature cotyledon shaped embryo will be formed. Dear students, in this part you see this part will be radicle and this part will be here, this side will be plumule. So the radicle and plumule, these are the structures present in the embryo. Radicle produces the root and plumule produces the shoot. Now cotyledons, these leaf-like structures are for providing nourishment to the developing plant, Now post fertilization, or here I have to tell you post fertilization structure and events. Number one, endosperm formation. Just like we have seen that in this diagram, you see this part, this red colored nucleus, it is a primary endosperm nucleus, and this is present in this cell. So this cell, this full cell will be known as primary endosperm cell. This is known as primary endosperm cell. So now these two things, that means primary endosperm cell which is having a primary endosperm nucleus, undergoes division, and this thing will produce endosperm. Okay, so is it clear? Now we see endosperm development and embryo development, maturation of ovule into seeds and ovary into fruits, everything will be included in post fertilizational events. Okay, in the post fertilizational, post means after, and fertilizational means fertilization, fusion of male and female gamete. So after double fertilization, post fertilizational events will occur. So the first thing what will be there, number one, endosperm development, and after that embryo. After that I should not say, actually after what you call first endosperm development starts. Okay, and immediately after that embryo development will start. Why? Because embryo requires some nourishment. So that nourishment will be provided by the endosperm. That's why first endosperm should be developed, and after that embryo should be developed. Now see here, primary endosperm cell, that is a triploid, that has that produces endosperm, which is again a triploid with reserve food. It is having reserve food, so it will provide nourishment to the developing embryo. Okay, so if there will be no endosperm, then how the embryo will be developed? Because embryo wants nourishment, embryo wants nutrition, so that nutrition will be provided by the endosperm. So first endosperm should be produced, after that, okay, or what you call simultaneously the development of embryo will also be started, but first endosperm development should be started. Okay, now primary endosperm nucleus, okay, that is triploid, this undergoes free nuclear division. Dear students, I want to tell you the free nuclear division. Just see this diagram. 1 minute. Just see. One second. Oh, I think it is not here, but I want to tell you, this thing, this diagram you see here, it is having two nuclei. This cell is having four nuclei. So you see here, two nuclei are making four nuclei, and 4 to 8. So nuclei are dividing, but cell is not dividing. This type of division is known as free nuclear division. So now once again coming to that part where we were, just see, sorry, so just come to this part. So here what will be there? The primary endosperm nucleus undergo free nuclear division. Okay, that means nuclei will be dividing, but actually the cell wall is not forming. So this is known as free nuclear division. After some time, the cell wall formation will be there. When the cell wall formation will be there, it is known as, now the endosperm will be known as cellular endosperm. Before that, it is nuclear endosperm. Okay, so I'm telling you, in coconut, the coconut milk is a free nuclear endosperm. That milk is actually the endosperm. And in that milk, thousands of free nuclei are present. Okay, but there is no cell. But in the coconut, white kernel of the coconut is the cellular endosperm. That white part of the coconut, that is actually cellular, but the what you call that milk, that liquid part, that is not cellular till now. Cell wall is not developed, so that is known as free nuclear endosperm. Now, endosperm may either be completely consumed by the developing embryo, that means embryo is developing, that can completely consume the endosperm, or some part of endosperm may be persist in the mature seed. Okay, so that means endosperm may be consumed completely in case of pea, in case of groundnut, in case of beans, in case of pulses, it will be consumed completely, okay, before seed maturation. But in case of castor and coconut, it may persist in the mature seed and be used during seed germination. Okay, so it is clear, dear students, you should not be confused with the endosperm and cotyledons. Cotyledons are the part of embryo and endosperm is present what you call around the surrounding the embryo. It is present what you call, I should say that endosperm is having the embryo, or embryo is present inside the endosperm. Okay, so cotyledons will provide the nourishment to the developing plant, but endosperm will provide the nourishment to the developing embryo. Okay, now, development of embryo. Embryo develops at the micropylar end of the this end of the embryo sac where zygote is situated. Okay, so here this type of structures will be there. This thing we have dealt already, so we are not going to tell you detail, but only we will read. Okay, so the what you call, where the zygote is situated, most zygotes divide only after certain amount of endosperm is formed. I have told why. This is an adaptation to provide assured nutrition to the developing embryo. Now, the seeds differ greatly. The early stages of embryo development, that is embryogeny, are similar in both monocotyledons and dicotyledons. Okay, dear students, just move in this. Development of embryo. See, the zygote gives rise to the proembryo first. What will be there? First proembryo will be formed, and after that it will be converted into globular, heart-shaped and mature embryo. Okay, globular embryo, heart-shaped embryo, and then mature embryo. Okay, such type of embryo is mature embryo. This is also mature embryo. Okay. Acha, this is the dicot embryo and this is the monocot embryo. What is the difference? In dicot embryo 1 and 2, these two are cotyledons, but in monocot only one scutellum is there. This scutellum is the cotyledon in case of monocotyledons. Okay, that means what is the another name of the cotyledons in case of monocots? So this is the another name. This is the another name. Which is the another name? Scutellum is the another name of the cotyledons of monocots. So now, a typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of the embryonal axis above the level of the cotyledons is the epicotyle. Actually, you must know this thing, it is very important. Actually, the portion of embryonal axis, okay, the portion of embryonal axis above the level of cotyledons is known as epicotyle, which terminates with the plumule or stem tips. Means this region, this full region will be epicotyle and this region will be hypocotyle. Okay, hypocotyle region produces the root. Actually here radicle is present, so because of radicle it will produce root, and here plumule is present, so because of plumule it will produce shoot. Okay, now the cylindrical portion below the level of cotyledons is the hypocotyle that terminates at its lower end of the radicle or root tip. The root tip is covered with the root cap. Okay, root tip is covered with the root cap. So now you must know radicle for root and plumule for shoot development. Now, types of embryo. Embryos of monocotyledons possess only one cotyledon, that is known as scutellum. I have told. In the grass family, the cotyledon is called scutellum, that is situated towards one side lateral of the embryonal axis. At its lower end, the embryonal axis has the radicle and the root cap enclosed in an undifferentiated sheath called coleorhiza. You must know this coleorhiza is the initial part of the root, you can say. Coleorhiza is the initial part of root which is developed from the radicle. The portion of the embryonal axis above the level of attachment of the scutellum is the epicotyle, and epicotyle has the shoot apex and few leaf primordia enclosed in a hollow foliar structure called the coleoptile. So from coleoptile, what you call shoot will be produced, and from coleorhiza root will be produced, because coleorhiza will be germinated from plumule and coleoptile will be generated, sorry, coleorhiza will be generated from radicle and coleoptile will be generated from plumule. Now, seed. It is the final product of sexual reproduction. You all must know. Seed is the final product of sexual reproduction. What is the definition? A fertilized ovule is known as seed. It is very simple definition. A fertilized ovule is known as seed. Seeds are formed inside the fruit. Seeds are formed inside the fruit. Okay, it is formed inside the fruit. Now, if it is a fertilized ovule is known as seed, then what will be the fruit? A fertilized ovary will be the fruit. Okay, this will be the definition of fruit. Now, it consists of seed coat, cotyledons, and an embryonal axis. What will be present in the seed? Three things: a seed coat, cotyledons, and an embryonal axis. Embryonal axis will have plumule and radicle, and coleorhiza, coleoptile, all these things. Okay, now the cotyledons of the embryo are simple structures, generally thick and swollen due to storage of food reserve, such as in legumes. Okay, in legume, the two, just like you have seen the gram, just like this. So here, if you will divide into two, the gram will be divided into two. It can be divided into right and left. So these right and left parts are cotyledons. Actually, this is not the endosperm, this is cotyledons. Okay, and this will provide nourishment to the growing what you call plantlets. Okay, to the germinating plantlets. Okay, now endosperm will provide nourishment to the developing embryo. Okay, now mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm. Non-albuminous seeds will not have any endosperm, or as it is completely consumed, endosperm is completely consumed during embryonal development. For example, in pea, groundnut, pulses. Okay, now albuminous seeds retain a part of endosperm as it is not completely used up during embryonal development. That is wheat, maize, barley, castor. Okay, in these cases, these are albuminous because they have residual endosperm. Some part of endosperm is left. Okay, so that endosperm will be used. Acha, if some part of endosperm is left, then cotyledons will be less. Cotyledons will be less matured. Okay, because here endosperm is available for providing nourishment to the developing plantlet. So cotyledons will not be developed in such a manner. Now, occasionally, in some seeds such as black pepper and beet, remnants of nucellus are also present, are so persistent. This residual persistent nucellus is known as perisperm. This is known as perisperm. Okay, so now just see, this is the structure of seed. Here these are two cotyledons. This is known as micropyle, you see, and this part. Okay, now this is what a seed. In the seed, you see the first thing is seed coat, then endosperm, then cotyledons. This is cotyledons. Now you see this region is hypocotyle region, and where the root cap will come out, and this region will be what you call here this region. Means below this will be hypocotyle and above this will be plumule region. Okay, so from where shoot will come out. Now you see this part. Here you see seed coat is showed, cotyledons is showed, and shoot, apical meristem, hypocotyle, uh, root axis and root tip and endosperm. This is a monocot. Okay, this is again a monocot. Okay, so see here coleoptile you see, plumule you see, radical you see, coleorhiza you see. This is what this is what you call, this one is of dicot and this is of maize monocot. So you see a pericarp is present. This pericarp I have already told, this will be the nucellus. This will be the nucellus. Persistent nucellus is known as pericarp. Now inside pericarp you will see endosperm, and this will be, this thing will be the cotyledon, that is scutellum. Okay, scutellum is a cotyledon in case of what you call this one monocotyledons. Okay, now coleoptile, plumule, radical and coleorhiza. That means radical makes coleorhiza and plumule makes coleoptile, and these two things will produce the stem as well as roots. That means shoot and roots of the plants. Now you see this is a flower, sorry, this is a fruit. Here you see thalamus is there. Okay, thalamus is there, so it is known as false fruit. It is known as false fruit. Here also the thalamus is there, so it is known as false fruit. Now we are coming to the next part, the seeds. See, the integument of the ovule harden as tough protective seed coats. Okay, the micropyle remains as the small pores in the seed coat. For why micropyle remains? It is for entry of oxygen and water into the seed during germination. Okay, so this you must know. What is the importance of micropyle in the seed? It actually facilitates the entry of oxygen and water into the seed during the seed germination. Okay, oxygen of water into the seed during the seed germination. Now as the seed matures, its water content is reduced and seeds become relatively dry. Okay, 10 to 15% moisture by mass. Okay, the general metabolic activity of the embryo slows down. Here the general metabolic activity of embryo slows down. This state of inactivity is known as dormancy, and either seed undergoes dormancy, that means it will be metabolically less active, or if the favorable conditions are present, that means adequate moisture, oxygen and suitable temperature, then the seed germinates. Now as ovules mature into seed, the ovary develops into a fruit. Okay, for example, the transformation of ovules into seeds and ovary into fruit proceeds simultaneously. Now, the wall of the ovary develops into the wall of fruit. The wall of ovary develops into the wall of fruit called pericarp. Okay, so here you see the wall of ovary develops into the wall of fruit, that is called pericarp. Now, the fruit may be fleshy. It may be fleshy just like in guava, orange, mango, or it may be dry just like in groundnut and mustard. Okay, now many fruits have evolved mechanism for dispersal of seeds. Okay, this thing will be known as cory, just like anemophily, just like we have seen in pollination, anemophily, hydrophily. In that manner, here the term will be used as hydrocory, anemocory, zoocory and so on. Now the fruits, true fruits will be those, in most plants the fruit develops from the ovary. Okay, only ovary. So if the fruit develops from the ovary, it is known as true fruit. Other floral parts degenerate and fall off, so this will be the true fruit. Now false fruits in case of, in a few species such as apple, strawberry, cashew, etc. The thalamus also contributes to fruit formation. So here what is there? Here only ovary is not involved. Some other part like thalamus is involved. So that is known as, that type of fruits are known as false fruit. Now parthenocarpic fruit. You see, listen carefully. Parthenogenesis is the thing in which we studied that seed formation without fertilization. Okay, and here we are going to study fruit formation without fertilization. So in this case, seed will not be formed. This is the difference between parthenogenesis and parthenocarpy. Okay, acha. In case of parthenogenesis, seed formation without fertilization, and here there will be no seed at all. Here there will be no seed formation at all, because here fertilization is not proceeds. That's why seedless fruit will be formed. Now, development of fruits without fertilization, for example, is banana. They can be produced by using plant growth hormones. Now, importance of seeds and fruits, this you all know, so I am not going in detail, but the seed and fruits, these are important for dispersal process, number one, and second thing, for avoiding that time period which is not favorable to germinate. So that time period they can overcome by making seeds and fruits. Okay, Acha. And this is the importance in agriculture also. Acha. One thing more, in the seed form or fruit form, they can they can make the what you call reserve food also. They can preserve the reserve food also for the germination, time of germination. So this thing also is important. Now, viability of seeds, just like viability of pollens we have seen, we have to see the viability of seeds also. Seeds will be viable up to a certain extent, up to at a particular temperature and particular moisture content, and so on. Various factors are responsible for this. Now, this is the last slide of this lecture. You see, apomixis and polyembryony. Okay, what is the name? Apo means without. Okay, so here we will study a few flowering plants, such as some species of Asteraceae and grasses, have evolved a special mechanism to produce seeds. Which type of seeds? Apomictic seeds. Apo means without fertilization. Apo means without, and mictic means fertilization, mixing. So without fertilization. Okay, so this thing is clear now. Apomixis is a form of asexual reproduction that mimics sexual reproduction. Apomixis is a form of asexual reproduction that mimics sexual reproduction. Now, in some species, the diploid egg cell is formed without reductional division. Means egg cell is diploid already. If egg cell is diploid already, then there is no requirement of meiosis or reductional division. So it develops into the embryo directly. Okay, means egg cell will not require any male gamete, and it develops into the embryo without fertilization. Okay, so this thing will be known as apomixis. Okay, you I think know about it, that how apomixis will proceed. Now, in citrus and mango varieties, some of the nucellar cells, you must know, I have told already, nucellar cells surrounding the embryo sac starts dividing, protrude into the embryo sac, and develop into the embryos. That means nucellar cells, these are producing embryos. In such species, each ovule contains many embryos. Each ovules contain many embryos. Okay, occurrence of more than one embryo in a seed is referred to as polyembryony. Okay, so I think this is also clear. Now, these two processes are involved in production of hybrid seeds. Okay, what do you mean by hybrid seeds? Little bit I have already told when I was dealing the artificial hybridization, so that is sufficient here. Now, what is the difference between parthenogenesis and apomixis? In case of parthenogenesis, both what you call the egg cell was haploid and male gamete was not involved. So here, by the parthenogenesis, haploid seeds will be produced. Haploid seeds will be produced. Okay, and in case of apomixis, diploid seeds will be produced. Okay, because here the egg is diploid. So this is the difference between these two things. Now I believe it is clear. So thank you. Enjoy online study. Thank you. If you have not subscribed my channel, please subscribe it. Thank you so much.